Hi, biology kiddos. So we are now doing part 2, which is membrane-bound organelles. So membrane-bound organelles are those organelles that were in more complicated cells. So we had prokaryotic cells, which were simple cells. Pro means before, so they became before the complex cells. And now we're going to be talking more about complex cells, which don't just have organelles, they also have membrane-bound organelles. So, non-membrane-bound organelles were cytoskeleton, ribosomes, cytoplasm, and the cell membrane, okay? Membrane-bound organelles are going to be enclosed in a membrane. That compartmentalization allows these cells to be more organized and more complex. All right. So, membrane-bound organelle number 1 is the nucleus. The nucleus is surrounded by a nuclear membrane. You can see that membrane right here. Inside of the nucleus, sometimes you'll see this dark spot. That's called the nucleolus. The nucleolus's job is to make ribosomes. Nucleoluses. All right, and then here you can see DNA. Now, DNA doesn't look like it's in its double helix form right here, but if I was to get a closer look, you definitely see that double helix. We call this loosely jumbled spaghetti-like DNA chromatin. Now we're going to talk about, so here's the nucleus. We're going to work our way out. This is endoplasmic reticulum. Okay, so endoplasmic reticulum, you can see right here, is going to be compartmentalized. It creates these little chambers, okay? It is both a transport system, it's an intracellular highway, so things come from here, they go into the endoplasmic reticulum, they get modified, they get exported, then they can go to a different type of endoplasmic reticulum, they can be shunted outside of the cell, they can go to the Golgi apparatus, so you're part of this transportation highway, usually for chemicals and products of the cell. Rough ER looks like this. It has ribosomes. And smooth ER is going to be like this. Do you see how this ER doesn't have the ribosomes? It's similar in look, but it's smooth on its surface. That has no ribosomes to make little tiny bumps, right? So it's smooth ER, and rough ER usually focuses on synthesizing and manipulating proteins, and smooth ER usually works on just synthesizing, manipulating, and moving non-protein materials in the body or inside of the cell. Okay, so next we're going to talk about the Golgi apparatus. So the nucleus contains instructions for everything that the cell needs to do and for you in general. And it's going to give those instructions to the endoplasmic reticulum, where it's going to synthesize and make whatever it needs to make, whether it's proteins, the rough ER, or whether it's other biological molecules in the smooth ER. Then it's going to be transported to the Golgi apparatus. The Golgi apparatus is going to take in the materials from the smooth ER and the rough ER. It's going to combine them together, repackage them, and then ship them either to inside of the cell using a vesicle, which is a transport vehicle of the cell, or it's going to ship it out of the cell, where you can see right here, the vesicle is going to bind to the cell membrane and push the contents out of the cell. So I think of Golgi apparatus as like the post office of the cell, and a vesicle is a cell vehicle where it transports things around the cell. Okay, the next one that we're going to talk about is lysosomes. Lysosomes contain digestive enzymes and they break things down. They will not survive the process. So sometimes people refer to them as a suicide sac inside the cell. Try saying that 10 times fast. But what happens is this lysosome has digestive enzymes in it, and so it will fuse with an old mitochondria that's worn and not working very well, or maybe a little bit of endoplasmic reticulum, and those digestive enzymes will eat up whatever it is. But because it's pretty much giving itself to the entire process, the lysosome is going to die in that process. So it just will bind to this part of the Golgi apparatus, digest it, and then those pieces that were used to build this Golgi are going to be broken back into their monomer components, so the cell can reuse those to build the polymers once again. Once again, that biochemistry unit, It's coming back. It's going to keep coming back. All right. So here is a young child that has Tay Sachs. She was born with defective lysosomes. Think about it for a second. If you were born with defective lysosomes, how would that impact your health? Right? So your cells wouldn't be able to get rid of old and worn out parts, correct? So would your cells be healthier or would they be sicker? They'd definitely be sicker, okay? Because they can't break down the waste, those substances build up inside of the cell and eventually the cells start to die, and that causes Tay-Sachs syndrome. Unfortunately, this is a disease that the baby's not going to survive. It's very, very sad, and they all die in early childhood. Lysosomes are also sometimes called the suicide sac because a cell that has become damaged or old and worn out, or it's just a cell that needs to die in order for the multicellular organism to mature, it will just release all of the contents inside of those lysosomes, and it'll pretty much digest itself and break itself down, and that is called apoptosis. Apoptosis, you can see right here, pop. Think of a popping cell, apoptosis. And the cell digests its insides first, and then its membrane kind of comes into itself, and then it just breaks down. And we call the breaking of a cell lysis. So once the cell completely breaks down, we call that the cell has lysed. Now, if I was to ask my students, what's your favorite organelle? Almost all of them are going to say, the mighty mitochondria, the powerhouse of the cell. The mitochondria is the cell's energy center. It turns food into a chemical called ATP. Now, ATP, adenosine triphosphate, ATP is a charging molecule. So if you think of like a charged battery that you can put into any of your electronic devices, ATP is like that inside of your body, right? So it can be plugged into a variety of different tiny machines, enzymes, inside of your body, and it can make them do work, right? Then it's turned into ADP. I think of ADP as the dead battery. So the D stands for dead battery. And this is the charging center. The ADP comes in, the ATP comes out. The ADP comes in, the ATP comes out. It comes to the mitochondria where it uses the energy that was stored in your food to recharge your batteries. And the charged battery is called ATP. That's why this is called the powerhouse of the cell, is because technically it is a complicated charging center. Another organelle that I want you to know is actually not membrane bound, but is in only complex cells. It's called centrioles. In fact, this is a very specific non-membrane bound organelle that is only found in animal cells. It's used during cell division, and it kind of acts as like a conductor. It helps to get things organized and helps the cell to know in which direction it needs to separate. Vacuoles are also membrane-bound organelles, but vacuoles are most commonly found in plant cells. So plant cells oftentimes will have a large central vacuole. It's where they store their water, right? Like if a plant gets thirsty, it can't walk to the stream and go get a drink. So it depends on storing lots of water inside of each individual cell in a vacuole. Sometimes animal cells will have small vacuoles. Fat cells have large storage vacuoles because they store lots of fat, right? Adipose cells or fat cells. But most of the time, animal cells will not have vacuoles, or they'll have very small ones. Whereas a plant cell is going to have this large, huge vacuole taking up most of its inside. So overall, you're noticing that eukaryotic cells especially are pretty complex, right? They have a variety of different organelles, and then they also have these complex compartments where specific jobs are done called membrane-bound organelles. And each membrane-bound organelle is like a space within the cell that can do one particular function or produce one particular product. So if you think about it, a cell is kind of like a factory. DNA has instructions to build, let's say, a protein. The instructions are sent to ribosomes. Let's say the ribosome is embedded on an endoplasmic reticulum. It then ships those proteins through the ER into the Golgi body, where it's going to package up those proteins and it's going to stamp that little package and be like, you need to go to this part of the cell, it needs some proteins over here, or it's going to say like, oh, let's secrete some proteins to assist in digestion, or something along those lines. So your cells are like complicated factories, right? You've got HQ, headquarters, with the instructions. You got to get those instructions to the workers in the proper order, in the proper compartment, at the proper time for the product to be produced for export or for cellular repair. Okay, kiddos. So I want you to study the cell, and I want you to be able to visually recognize each individual part, right? So you need to know that this is the nucleus. That's a nucleolus. This would represent chromatin. This is a lysosome. What's that fluid called? The cell soup? Oh, that's cytoplasm. These are little free-floating ribosomes, but those are ribosomes embedded into this endoplasmic reticulum, which makes it rough endoplasmic reticulum. This would be smooth endoplasmic reticulum. The stacked sacs over here are going to be the Golgi apparatus. These little weird sticks are supposed to represent cytoskeleton. I don't know, not my favorite picture, but it works. It does its job okay. These are the mighty mitochondria. They're going to be charging up those ATP. And I believe that you guys remember these centrioles are used to help cells divide. Okay, so now what I'd like you to do is I'd like you to label this diagram. Pause. Kiddos, remember when we stained your cheek cells and you looked at them underneath the microscope? Let's look again. This is a pretty good picture I got of one of your stains. And let's think about what we're seeing here. So first of all, this dark spot in the center of the cell, those are going to be the nucleus, okay? These little spots right there, those are mitochondria, okay? Those are the powerhouse, the charging centers inside here. Now, I'm not sure if you guys can you see the ruffle stuff right here around the center of the cell? Let me see if one of them is a little bit more, like you can see it really well right here. You can see that this would have been a nucleus. And then right here is that ruffle stuff. That's endoplasmic reticulum. Now, ribosomes are too small for us to see. And actually, like, I can't tell the difference. Like, this is a mitochondria. This might be some sort of vacuole, or it could be some sort of lysosome or something. The mitochondria are going to be pretty standard sized. And there's lots of mitochondria in this one, right? Do you guys see that? Okay, so this is a cell. We're able to see cells underneath the microscope. We can see your own human cells underneath the microscope. So it's really important that you understand the parts of the cell and you know what each part does. And because we're animals, it is really important for us to understand the different cellular parts for animals. But plant cells are similar, yet different to animal cells. They do have a few structures that I want you to be familiar with, and I want you to be able to recognize the difference between an animal cell and a plant cell. Here you can see that large central vacuole that I was talking about before. That's going to store water and nutrients for the plant. Cells also, plant cells don't just have a cell membrane like animal cells do. They also have a cell wall. That cell wall helps give plants structure because they don't have a skeleton like animals do, okay? In addition to that, although cells have mitochondria, they can break down sugar and use that sugar to charge ATP to run cell machines, okay? Although they have mitochondria and they can do that, they can also make their own sugar from sunlight and CO2, and that happens in the chloroplast. That process is called photosynthesis. When plants can take sunlight and CO2 and turn it into sugar, that's photosynthesis. Some of that sugar is going to get stored in this large central vacuole. Some of that sugar is going to be used to charge molecules so that the cell can build its body. And sometimes some of that sugar is going to go into fruit or something that we can eat and that we get our energy from. Okay, kiddos, now can you identify all of the parts in this plant cell? Make sure that you label each. Go ahead and pause. So this is Elodia. It is a type of aquatic plant, and it's fairly common. It actually is a little bit invasive here in Florida. Oopsies, okay, from the aquarium industry, got let loose. But if you looked at the plant cells underneath the microscope, you can see these little green dots. What do you think these green dots might be? If you said a chloroplast, you are right on. And so that's the first thing that we look at when we're looking at plant cells. We're like, can we see those green dots? Because that's a good indicator that we're looking at a plant cell, okay? So if I saw this, I would note, okay, well, I'm seeing all these green dots. It looks like there's a dark area in the center, probably the nucleus. These are actually closer to the surface of the cell. It's not focused all the way through. That might be a nucleus right there. But I'm going to look for green dots when I'm looking to see, is it a plant cell? Is it an animal cell? Animal cells are not going to have those large green chloroplasts. When I'm trying to determine the difference between a eukaryotic cell and a prokaryotic cell, I am going to look to see, is there a membrane-bound organelle? You can see that pretty clearly underneath a microscope. Remember those cheek cells? You can see that dark purple spot pretty clearly. If it has a dark purple spot in its center, then it's probably a eukaryotic cell. You don't even have to identify the other membrane-bound organelles. If it has a nucleus, it's a eukaryotic cell. If it doesn't, it's probably prokaryotic. Plus, prokaryotic cells are usually much, much smaller than eukaryotic cells, but we're looking for a nucleus. So you know how I said that mitochondria and chloroplasts have these membranes, right? And I even pointed out that like a mitochondria, it has a double membrane. Do you remember me saying that? Okay, so chloroplasts do too. That is because mitochondria and chloroplasts, we're pretty sure, used to be their prokaryotes once upon a time. So what we think happened is, remember in that diagram, that little video where I showed you the amoeba eating the prokaryotes? We think that there was large cells that ate smaller prokaryotic cells, and instead of actually digesting them, it was like, oh, hey, you're making lots of sugar. Okay, well, I'm not going to digest you then. I'm just going to go ahead and live with you in a symbiotic relationship. Tell you what, I'll give you some protection. You give me some sugar, Mr. Man, right? And eventually, over time, they became so reliant on each other for survival that they became like one organism. Kind of like we have symbiotic critters that live inside of us. Like we have bacteria and things that we can't digest food without. So we would die without them, right? So eventually, over time, these guys became so synonymous with the cell that we called them one organism. But mitochondria and chloroplasts, not only do they have their own membranes that have proteins that are associated with prokaryotes and not eukaryotes, but in addition to that, they have their own DNA. They divide themselves. So they're like little critters that are just living in this beautiful symbiotic relationship inside each cell. That idea that these guys were ancestral prokaryotes that have become ingested and then symbiotic with cells is called the theory of endosymbiosis, meaning it's a symbiotic relationship, but they are within the cell. Okay? Now remember I said that cell structure is related to function. You can see here that different cells look incredibly different. Okay? For instance, this is going to be digestive tissue, right? You can see these little tiny villi that are sticking out of the surface of the cell. They're increasing surface area, so as food goes through digestive tract, there's more area for the food to absorb, right? This would be a tissue that's going to be in a skin or like a lining inside of your, let's say, esophagus or something. So it's going to create a barrier that separates your esophagus from other tissues. This right here, I think you guys know, is a neuron, right? So it's got these long tendrils which help it talk to other neurons. And this right here, these are adipose tissues. Remember I said adipose cells or fat cells are weird because they have big vacuoles and no other animal cell does? Yeah, they're filled with fat. They're like nuclei is like squished off to the side here. Do you guys see that? That's an adipose cell. All right, kiddos, this wraps up part 2 and our little foray into complex eukaryotic cells, the differences between animal cells and plant cells, the difference between prokaryotic cells and eukaryotic cells, the membrane-bound organelles, and the job of each. Thank you for sticking with me. I know it was a long lecture, and I appreciate you. All right, good job, kiddos. Our next lecture, as I'm sure you're guessing, will be cell membranes and homeostasis.